My name is Sergio Baranzini, and I'm a professor in the Department of Neurology at the University of California in San Francisco, and I am the Barancik Prize winner. MS is an autoimmune disease, so immunology is central. MS is a genetic disease, so genetics is essential. We use sophisticated imaging to diagnose MS. We need expert clinical and neurological examination. No single team will likely solve MS forever, and we are very excited to be part of this conversation and to be part of the solution.
I've had the pleasure of collaborating with Sergio Baranzini for almost 10 years here at UCSF. One of the striking things about Sergio is just his tenacity in pursuing questions. He's been working in the field of genetics for a long time.
From the early 2000s until today, we've identified more than 200 associations with common DNA variants that contribute to MS susceptibility. Now, in recent years, we've turned our attention to the issue of disease progression. What we discovered was that indeed there was one DNA variant in chromosome 2 that confers a much higher risk of progression. We think that identifying these variants will set up the stage for developing drugs that can address the issue of severity and progression. Our initial studies of the genetic contribution to multiple sclerosis risk taught us a lot about exactly that, but also taught us that genetics, as important as it is to determine the risk, it may not be the complete answer. So we thought that trying to understand the composition of gut microbes will tell us the environmental triggers of multiple sclerosis. And one of our earlier studies, we sequenced DNA from gut samples from MS patients and compared those to healthy controls, in fact, to their partners. And we found significant differences in the composition of the gut microbiota. That was really eye-opening to us.
One of the things that's unique and innovative about Sergio's research is that he is able to leverage these massive databases. His Spoke knowledge network, sort of like the database of databases, is able to pull together massive amounts of data in a way that we can actually use to ask specific questions. How is this symptom related to this medication, to this gene?
Spoke has been utilized, for example, to analyze millions of electronic health records of individuals who will develop MS in the future. And then by comparing them to individuals who never develop MS, can the machine identify individuals who are at risk of MS? And the answer is yes, we can identify them even several months or even years before the disease manifests. The Barancik Prize is an incredible honor. It really takes a village for the kind of research that we do. I'm extremely grateful for the faith and the support that the MS Society has, not only for me, but also for young scientists who are promising. They literally shaped my career from the very beginning. A cure for MS will mean a lot. It would mean that the lives of people who lived with MS will be transformed forever. That is possible. It's possible to live in a world where MS is not part of it, and I'm really glad to be working every day towards that.